Yeah, so I would like to thank the organizers for this kind invitation. Yeah, so I'm working at Natera as a machine learning research scientist for the past 1.5 years, leading the Genomic Foundation Models efforts. And today I will talk to you about Genomic Foundation Models and AI and how can we use these type of models to advance precision oncology. So just to give you a brief background for the company, so Natera is a diagnostics company and is considered as a global leader in cell-free DNA testing. This means that we analyze small, tiny DNA fragments that exist in a simple blood samples. So this has huge implications in various domains such as oncology, women's health, and organ health, as we can use these tests to detect disease early and monitor the patient health. Uh, Specifically, we process millions of tests worldwide, and our flagship product, Signatera, is used to for personalized cancer monitoring. So on a high level, Signatera basically sequences the patient's own tumor. We detect the specific tumor mutations, and then basically we develop a personalized blood test that can detect the tumor DNA in the blood sample. So basically, doctors use this test even earlier than standard medical or imaging tests.
Not working, so
Yeah, so as we gather more and more data, we believe that AI can unlock the next innovations in precision oncology, and I will explain why. So we believe that cancer biology has a hierarchical nature. So starting from the DNA mutations, these effects propagate up to the RNA, to the protein level, and finally they affect the patient outcome. So most models today, they operate on these single individual modalities, while here at Natera, we have the unique advantage that we process a huge multimodal data set. So having more than 300K cancer patients with whole exome, whole genome sequencing data, along with the ctDNA monitoring from our Signatera tests, paired with H&E histopathology images. And all of these, they are tied together on the patient level, along with the clinical history and other treatment and metadata information. So the goal is basically to leverage all of these different modalities and build foundation models that can extract information from the individual modalities and then to have a significant clinical impact. Specifically, we use these type of models to improve the monitoring of cancer patients, basically to identify high-risk patients and intervene earlier. Then we can use these type of models to identify new biomarkers, to design and find new targets, and also to match our patients with new treatments and new clinical trials. Uh, so in the first part of my talk, I will discuss about our internal DNA foundation model, specifically for cancer genomics. So you're all familiar with large language models. They have been trained on general internet text and they are optimized for natural language tasks. So people inspired from the success of LLMs, they try to adapt and apply the similar techniques in biology. So that's what we saw with genomic language models, basically big foundation models trained on biological sequences such as DNA, such as nucleotides in a DNA level or amino acid sequences, etc. However, these generic genomic language models, they are not specifically trained and designed for oncology tasks. So that's what our Natera's DNA Foundation model is doing. So we pre-train this model on many real-world cancer patient data, along with their clinical information. And we built this model specifically for oncology. So to give you more details for the pipeline that we used, so the input to the foundation model is both the genomic data, so this is the nucleotide sequences of the patient, along with some clinical information such as the specific cancer type, the age or the sex of the patient. And then in the first phase of the training, we have a standard next token prediction objective. So similarly to standard LLMs, we pre-train the model to accurately predict the next nucleotide in the sequence. And the motivation behind this pre-training stage is that we want to teach the model the underlying biological vocabulary and language. So this is the first step, and now you may wonder, like, why don't we just scale this type of models, collect all the relevant data that we have, and then we can have a big success as we see in standard LLMs. However, there are some differences in biological data versus in natural language text. And one of the main differences is that there is a small low diversity in genomic data. So if you compare the DNA sequences of two patients, the similarity is extremely high. So you can see that our task is much more challenging since we want the model to identify these very small differences, these very small mutations that can have a huge effect in the patient outcome. So that's why we believe that the standard paradigm of next token prediction is not enough for these foundation models. And that's where we introduce an additional post-training phase, specifically using a contrastive learning objective, using more than 140K patients, where the underlying objective was to redesign the latent space of the model such that clinically similar patients are grouped together in the latent space, while patients that don't have similar clinical features, they are pushed further away. So combining these two training strategies, we have a pre-trained model that can be used in some various downstream tasks. We can think about two main categories of the downstream tasks. In the first one, we just want to extract a prediction for a specific sequence, for a specific DNA sequence. So this one relevant task is, for example, a variant interpretation, where we want to interpret and predict oncogenic variants, while the second group of tasks are based on the patient level, where we integrate multiple DNA sequences from a single patient, and we learn a patient embedding where we use it for some other applications, such as risk prediction, etc. Great. So we basically evaluated our model on oncogenic variant prediction task, and we compared our foundation model with state-of-the-art DNA LLMs such as EVO2 and DNA BER2. So we see that our model is able to classify oncogenic variants with a great performance, often outperforming state-of-the-art models right now across the industry and academia. While we did an additional unsupervised evaluation here, where we extract the embeddings that the model learned from various sequences, and then we measure the quality of these embeddings using some linear algebra metrics. So here we see that Natera's DNA foundation model achieves a great rank me score, often outperforming other foundation models. Moving beyond this evaluation, we further wanted to see if our model learns some meaningful biology in the unsupervised embeddings. So here on the left side, you can see a Disney visualization of the embeddings of the model for different genomic regions, such as coding sequences, promoters, etc. So here we can see that our foundation model accurately creates a latent space with distinct clusters for these different genomic regions. While on the right side, you can observe the classification accuracy using a KNN classifier on top of these embeddings. Again, here, the model didn't see any of these labels during the pre-training or the post-training phase. Moving on to the most interesting translation applications that we work on, we have a hypothesis that the cancer-specific embeddings that we learn can be used in order to predict patient-level molecular risk. So basically, we have three different pipelines, three different tasks that we apply our model. So on the left side, you can see a single gene patient risk stratification. This means that we focus on a single gene. In this case, the EGFR mutated gene for lung cancer patients. And we basically significantly stratify the low versus the high risk patients just from the embedding of the specific gene that we learn. Secondly, we applied some in silico mutagenesis techniques. So basically, we applied different mutations in the gene, and we observed how the predicted patient risk increased or decreased. So there you can basically, we can see that we can identify some drug resistance mutations using the gene embedding that we learned. And finally, we focus on a multi-gene patient stratification setting where the input is basically multiple genes with all the different mutations from the patient. And then we use all of these embeddings in order to, again, stratify between low and high risk patients, achieving, again, a significant stratification. So specifically for this third task, this is the pipeline that we used. Starting from the input construction, we basically extract the different somatic variants and we construct the transcript sequences for each patient. So this gives us basically a set of sequences, a set of nucleotide sequences for each patient. Then we use our foundation model in order to embed each sequence separately, followed by a pooling aggregation layer where we combine all these different foundation model embeddings into a single patient-level representation. Then we use this patient-level representation in order to predict the final risk score and train the model end-to-end using survival loss. Now, moving on to the evaluation, basically we applied this model on colorectal cancer patients. Specifically on the left side, we can see the evaluation of stage 1 patients where our model achieves a significant separation with a index of almost 0.7 between low and high risk patients, while we also achieve a separation for the stage 2 patients as well. And we can see that the high-risk patients show consistently worse outcomes. So afterwards, we also compared our model with a standard clinical baseline model that uses only some clinical features without the foundation model embeddings. So basically, we extracted the predicted low-risk patients from the clinical baseline model, and then we used our foundation model to further separate these low-risk patients. So on the right side, we can see that, again, we have a separation between the low and the high-risk patients. So therefore, our model was able to identify patients that were predicted as a low risk from standard models, but they may need some closer monitoring in the end. So now we'll move on to the second part of my talk, which is about deep antigen sets. It's basically a new deep learning framework for effective targeted immunotherapy design, such as personalized cancer vaccines. And we published this work last year at AACR. So to give you some context the goal of personalized cancer vaccines is basically to enable and activate the immune system in order to recognize and destroy the cancer cells by targeting specific neoantigens. So these neoantigens are caused from specific tumor mutations, and they are these unique flags of cancer cells. However, this is a very challenging problem because, first of all, there are thousands of different mutations in a tumor, while only a tiny fraction of those is truly immunogenic. And moreover, we have limited experimental validate data. So we cannot efficiently train AI models for that. Moreover, this is a very challenging and complex biological process because in order to have a neoantigen, you have to start from the different DNA mutations. Then these specific proteins should be expressed. And moreover, the specific small part, this mutated peptide should bind to the MHC molecule, and then the MHC peptide complex should basically presented in the cell surface, and then the T cells should recognize this complex. So there are these different steps that should all happen in order to have these neoantigens. And you can imagine that building an AI model that can accurately predict all of these steps is very challenging. So we basically modeled this problem in a slightly different setting, where we try to leverage our in-house patient data. So specifically, we had a labeled data set with patients that responded to IO therapy, and we created these positive and negative samples, along with the specific candidate neoantigens that each patient had. So basically, we trained a model by modeling the whole set of neoantigens of each patient, and we used a deep sets approach where basically the input is a set of neoantigens, and the output is the predicted score for the specific patient. So basically, if that patient responded to IO therapy or not. So once we train this model, then we can have different kind of applications. For example, we can use deep antigen sets in order to predict for new patients if they will respond to IO therapy or not. But also, we can use the same exact model to rank different neoantigens, and this can be very important in developing personalized cancer vaccines. Specifically, in our internal validation, we achieved 69% of AUC in predicting the clinical response of our in-house patient data. And also, our model was able to retrieve more than 60% of the true neoantigens in the top 20 predictions of the model, while the model was also able to retrieve more than 90% of the true neoantigens in the top 100 predictions of the model. And we validate our neoantigen prediction model in NCI Tesla and high-tide data sets, which are considered very important in our domain. So yeah, to summarize, I talked about our Natera's DNA cancer foundation model, specifically designed for oncology, trained on cancer sequencing data and used for molecular prediction, biomarker discovery, achieving superior and competitive performance against other state-of-the-art DNA language models. Moreover, we discuss about neoantigens, how we can use our in-house deep antigen sets model to predict IO response for patients, and also to discover and predict new neoantigens. And all of these as a next step, they should be integrated all together. So we are currently working some multimodal models as well. So this is the next big step. How can we combine all these different modalities all together to improve the patient representations that we learn from the foundation models? And of course, all of these, they should be backed up for real-world clinical data sets at scale. Finally, yeah, so this is my last slide where I would like to discuss a bit about this debate. So some people argue that LLM agents are what we need, while some other people, they mostly rely on these smaller specific bio models, bio foundation models. So we believe that there is a strong synergy between these two type of systems, where we can imagine a multimodal system that is using an LLM agent as the orchestration layer. And then these biospecific foundation model tools that I described previously can be used as tools for this LLM brain. And you can imagine that, for example, the LLM agent can design new experiments, call the specific biofoundation models, get the output from the model, and then have a refinement and improvement of the models as well. So this is the next step and the goal for our systems. Thank you very much, and happy to answer any questions.
Any questions to Michalis' talk? Yes.
Thank you for the nice talk. About the prediction of drug response with the multimodal data, would you be able to share like initial result? Is there like a particular modality which is much more important than the other, or is it like shared?
Response, you
Yes,
said?
patient response.
Yeah, yeah. Actually, we have models using imaging data. We have models using DNA data, models using RNA data. So far, we see that imaging and RNA are the most informative modalities. I think this is what other companies have shown as well. So we have a consensus right there. For the DNA models, it's a bit more challenging. I think one of the reasons is this low diversity that we have in the genomic data, specifically for DNA. So I think that's the most challenging modality so far.
Some other question? Yes, over there.
Thank you. I'm just interested in how do you handle the three different reading frames you have for proteins? Because if you have DNA, a DNA region, it can be like coding on one frame, but the other frames are not coding. So is it intogenic? Is it, so how do you include it in your model?
Yeah, so basically, in most of the downstream tasks, we just use the coding sequences as input. So I just showed this experiment where we use other regions of the genome just to show that our foundation model can further separate these different regions. But for all the clinical and downstream tasks, we just use basically specific coding regions nearby the mutations that we identify for each patient. So we have a window across the specific mutation.
Thank you.
Is there any other question? I have a small question, Michali. It is about the data. I guess the data that you are using and experimenting on are private, right?
Yes. Yes.
Yes. So it is evident that in the absence of public data, such results cannot be verified, cannot be reproduced. How do you treat this issue? This is a bigger discussion, of course, but
Yeah, I mean, yeah, that's a fair point. That's why we basically try to always validate our models in some external and open source data, like in TCGA, for example, and see if our models can generalize to external cohorts, etc. So yeah, like always we start with our own data, seeing if our models work there, but then you have this external validation that you have to verify that your model doesn't just overfit in your cohort distribution.
Right. Thank you.